Welcome to another lecture by MedicoMedics. Cell Biology, Chapter 4, Cell Signaling. In this lecture, we will discuss why cell signaling is important, components of cell signaling, different types, receptors and their types. We will discuss the signal transduction pathways, major signal transduction pathways, regulation of cell signaling, disruptions, and end with a summary. So why is cell signaling important? Well, it plays a role in coordination of activities, so it ensures cells work together for growth, repair, and immune responses. It's important in response to the environment, so cells adapt to change like nutrient levels or stress. Regulation of processes, so controls essential processes like metabolism, cell division, and apoptosis. Clinical relevance. Disruptions in signaling can lead to diseases like cancer, diabetes, or autoimmune disorders. So imagine if a factory's workers couldn't communicate. Nothing would get done. So that's what happens when cells can't signal. Now let's explore the key components involved in cell signaling. So, one component is signaling molecules or ligands. These are chemical messengers like hormones or neurotransmitters and cytokines, etc. And if you look at this illustration here, you see the ligand in blue. So any molecule essentially binding to a receptor. That leads us to receptors that are proteins on the cell surface or inside the cell that bind to these ligands. When that has happened, we then move on to signal transduction pathway. That's a series of steps converting the signal into a response. And then we have the target cell response, which could be, for example, activation of an enzyme or changes in the cell's behavior or gene expression, etc. So a ligand binds to the receptor. We have a relay of the signal leading to a response. So you can think of signaling like receiving a message, right? The ligand is the letter, the receptor is then the mailbox, and the response is reading the letter and then acting on it. Now let's examine the different types of signaling cells used in communication here. So types of cell signaling. We have autocrine signaling, where the cell sends signals to itself. Examples: cancer cells producing growth signals. So autocrine is like talking to yourself. Then we have paracrine signaling. This is when signals target nearby cells. Example can be neurotransmitters in synaptic signaling. Then we move on to endocrine signaling, where signals travel long distances via the bloodstream. Examples include hormones like insulin. And then we have direct contact, or juxtacrine signaling, where cells communicate through direct physical contact. Example is immune cells interacting with infected cells. Now let's focus on how these signals are received by the cell through receptors. We have membrane receptors. These are found on the cell surface. They bind large or hydrophilic ligands. If you remember the head, right, we had the cell membrane made of a phospholipid bilayer, right? And we said that the head was hydrophilic, the tails were hydrophobic. So the ligands on the cell membrane are therefore hydrophilic. Examples include G protein coupled receptors or ion channel receptors. So that's membrane receptors. Now we have intracellular receptors. These are found inside of the cell, and these bind small or hydrophobic ligands like steroid hormones. So receptors are like locks, right? And these locks open only if you have the right key, which here is the signals. Now, after this signal has been received, the cell has to process it. So let's see how this happens in signal transduction. Now, the definition of signal transduction is the conversion of an extracellular signal into an intracellular response. Like we said, we had the cell membrane, we had a ligand binding, right? So it's an extracellular, it's outside of the cell. And it's supposed to convert this into an intracellular response. Now, the key steps include, first of all, reception. So the ligand binds to a receptor. That's step one. Or look at this illustration here. Here is step one. Then we have transduction, signal transduction, which is a cascade of molecular interactions amplifying the signal. Right? The process here. And then finally, we will have the response. So specific cellular activity like gene expression or enzyme activation. An example can be epinephrine, right? Epinephrine binding to a receptor can trigger glycogen breakdown in liver cells. So if this was a liver cell and epinephrine binds to a receptor on its cell membrane, glycogen could start breaking down. That's a perfect clinical example of signal transduction pathways. Another pathway is the major signal transduction pathway, or pathways. We have, for example, G protein coupled receptor pathway. It involves activation of G proteins and second messengers like cyclic AMP. Example of this is hormone signaling. Another one is receptor tyrosine kinase pathway. It triggers phosphorylation cascades. Example of this is insulin signaling. Another pathway is ion channel pathway. It opens or closes ion channels in response to signals. Example of this is nerve impulses. And each pathway is like a different road leading to the same destination, which is essentially getting the cell to do something. Now let's see how these signaling processes are regulated to prevent chaos from occurring. So when discussing the regulation of cell signaling, we have signal amplification. Small signals produce large responses, for example, cascade effect. Now we have feedback mechanisms, whereby we either have positive feedback, where the signal is enhanced, or we have negative feedback, which reduces the signal to maintain balance. Finally, we have signal termination, so mechanisms like receptor inactivation or degradation of signal molecules will terminate the signal. So cell signaling can be compared to a thermostat. It can amplify signals when necessary, but turns them off to avoid overheating. But let's look at what happens when signaling goes wrong. So in different pathologies, cell signaling has been disrupted. For example, in cancer, where overactive growth signals, or we have faulty tumor suppressor pathways. It's either one or the other. If it's overactive growth signals, the cell will continuously divide and produce more cells, leading eventually to a tissue, leading to the formation of the tumor. Or we have other pathways that are supposed to signal that these cells are not supposed to be dividing. Like we have tumor suppressor genes, for example. If they are not functional, if they are not signaling the way they're supposed to, we won't have the tumor suppressor in place. So this overgrowth can also keep occurring. Another example is diabetes, where we have impaired insulin signaling. We can talk about neurodegenerative diseases, where faulty neurotransmitter signaling occurs in conditions like Parkinson's. And we can mention autoimmune disorders, where overactive immune signaling lead to tissue damage. So, in summary then, when signals misfire or don't get through, the whole system could break down, leading to different diseases. And here we mentioned 4 different types of diseases. So in summary then, cell signaling is essential for coordination, adaptation, and regulation. It involves ligands, receptors, transduction pathways, and cellular responses. Disruptions in signaling are at the heart of many diseases. Understanding signaling pathways provides targets for drug therapy. And hopefully now it starts becoming more and more clear why we study cell biology and why it is so important. Because signaling is an essential part to the function of cells. And as we just mentioned, if these signals are not working as they're supposed to, we could have, for example, overactive growth signals leading to a disease like cancer. Or we could have diabetes, or Parkinson's, or autoimmune diseases, etc., etc. And that's the end of chapter 4. Continue now to chapter 5.